I'm not Farica. I think you can tell. I'd like to thank the organizers for selecting Farica's work. I'd like to not thank the bureaucrats that didn't get her a visa so she can come and present. So I was asked to do that. And so I'm going to talk about Farica's work. Uh, she's over there. Okay, so let's start. So this is about RNA G-quadruplexes. G-quadruplexes are structures that are formed between short tracks of Gs. As you can see here, it can form in DNA, it can form in RNA, but we're only going to talk about RNA because we only care about RNA. To be fair, this is the canonical version, but there's also non-canonical versions, like ones that only have 2G repeats or G triplexes, etc., that are shown here, and we'll talk about them later in the talk. So where do they form? There are mostly in UTRs in RNA. Some of them are also in coding sequences, 3' and 5' UTRs. And this is the distribution of their locations that have been detected, and they can affect a lot of things: translation, alternative polydentilation, splicing, etc. So they can have a lot of functional roles. How do we detect them experimentally? There's two main methods that we'll mention today. One of them is circular dichroism, which is a spectroscopy method. You can see here the x-axis is wavelength, and you can get different measurements, different temperatures, and you can add ions, potassium or lithium, and see the changes in the spectrum, and you get a characteristic curve for their formation. Another way is to, again, add potassium and lithium, and that stabilizes the structure, and then you can do reverse transcription, which is going to get stuck where the structure is formed. You can compare with or without the ions, and you can get the locations of the formation of the structure. So these are the two main methods that we're going to talk about today. And so one of them was done on high throughput measure, this RT-stop that I just mentioned. And this was a paper that was published, as you can see, in 2016, reporting widespread formation of RNA G-quadruplexes in the human transcriptome almost at the same time. Bartel's group published in Science the claim that they do not form, right? Those two opposing views started what's now known as the RG4 wars that affected millions of RG4 researchers across the world. And so we came to this much later and said, let's look at this from a different perspective. Let's use population genetics and statistical genetics and see what genetics has to say about this, okay? So we published this paper in 2020 showing that indeed we see very strong negative selection on RNA G-quadruplexes. Doesn't mean that they form or not form structures, but we can see their functional importance. The main two take-home messages from this, I'd encourage you to read this, are that there is this negative selection, and disrupting them can affect genes in terms of expression, and we can see associations with GWAS signals, and etc. But there were limitations to this work because we only looked at canonical G4s and those that were reported with this high throughput RT-stop method that I mentioned, which were a few thousand hits in HeLa cells. So it's very limited, okay? So the question is, can we extend beyond there? And of course we can, and we're going to use deep learning in this case. So we're going to give a predictive model that will give us many more hits if we can get it to work accurately. And so what we do is we pre-train a language model, foundation model, use whatever buzzword you want to use, on over mRNA sequences. This is a BERT model with some extensions. I'm not going to go into the details, but once you do this, you can then fine-tune it. And on this about 10,000 sequences, positive and negative, and then you can predict both the formation RG4s and the subtypes that I mentioned before, canonical and non-canonicals. So how does it work? Here is a comparison to a convolutional neural network that was previously published. And you can see on test data that it performs better, slightly better on predicting the existence of RG4s, but much, much better in subtype predictions. Okay, so next we wanted to look at data that comes from other experimental methods. So this is a database, G4RNA, from Michel over here, and it has a few hundred sequences done by different experimental methods that you can see here on the x-axis. Okay, so we broke down this database. Again, these are different experimental protocols to see performance. And again, we compare G4mer to other methods, and you can see that it performs better. And I'll point you to two main methods here. One of them is luciferase assays, right? So that's allows us to measure potentially the entire UTR, longer sequences, more native basis circular dichorism, which usually uses very short sequences because that's what works with this method. And so that got us intrigued. And then we took the same data set and we broke it down by the length that was validated. And so you can see here high enrichment for very short sequences in which most methods tend to perform very well. And that would make sense if you think of it as this sequence is like 10 bases, 12 bases long, 15 bases long. You're going to have your tracks. You're going to have like maybe one position between it. And that's kind of it. But as you can see, as sequences get longer, then you can see much more differences in terms of predictive power, right? Because then you can get much more flexibility, non-canonical, and etc. So that made us think, okay, but what do we see in humans? Okay, and this is the interesting distribution that we found, right? So you can see here 3 curves for canonical RG4. So they follow the canonical one, and you can see very big differences in those distributions. The ones by RG4-seq is similar-ish to what we see in the 5' UTRs in humans, and the CD is way shifted, right? That's sort of maybe a lesson or a warning for us, because what happens, if you think of it, is people who use circular dichorism like to use short sequences because that's when you can see the signal, but it doesn't necessarily mean that that's what's actually happening in humans, right? So there's some lesson to be had here once you start looking at what your models are learning and where differences arise. Okay, and we'll get back to length later to see effects on function. So then we went back to population genetics and we said, let's look at allele frequencies in NOMAD and see whether there's a difference. And we're looking at variants that are breaking or not breaking the PG4s. PG4 stands P for putative. And so you can see here clear differences, statistically significant. And now you can say, oh, maybe you're hitting the Gs or not hitting the Gs. So now we're subsetting it only on variants that hit G-runs, either breaking it or non-breaking it. And again, you see a very clear trend of differences. So again, negative selection, again, breaking them. And these are all predictions from this deep learning models, right? So all of them go through the deep learning model and it's predicting breaking or non-breaking, right? So that's the game we're playing. So then we looked at short versus long. Coming back to my previous point, you can see differences, but they're not in the same direction. Now, when you just focus on the G tracks, you can see very strong differences, and they are in the same direction. So then there's the question of whether those are also functional in terms of what we see in terms of phenotypes. So turn to the Penn Medicine Biobank. That's about close to 50,000 now participant, where we have all exome sequences and all their electronic health records for all their phenotypes. And we focused, it's highly diverse, and we focused on the African and European population. We do some exclusion, as you can see here on the slide, for some variants and related individuals, etc. Then we look for associations. Now, of course, we can do this across all individuals, all variants, but we focused, also we want to validate on a specific subset. So we picked breast cancer, because as we know, there's a lot of variants of unknown significance for breast cancer, which are important for diagnosis and giving answers to patients. So we wanted, and a lot of those variants are in UTR, so they're basically ignored. So we wanted to look at this. So we took a known list of related genes, but also we did PheWAS, Phenome-wide association analysis for breast cancer. Here are some examples. So this is in a gene EPN3, which was not on the list that we got. We see genome-wide significance, specifically for breast cancer for an RG4 breaking variant. And when we looked it up, we actually found multiple publications implicating this in breast cancer, although it was not on our list, which was nice. And then we did dual luciferase assays. This was done by Danielle Gutman, who's here, is a postdoc in the lab. She did all the experiments. And you can see very clear effects on protein levels when we compare wild type to the knockout. And these are 3 different cell lines. And here is another example. This one was MHH6, so this was on our list. We couldn't get power enough in the Penn Medicine Biobank for the variants that we found, but we knew it was already implicated, so we tested it. And again, you see very strong effect on proteins when you introduce the mutation. In this case, it's an RG4 forming variant, right? So it goes in the other direction. Okay. And here is another example of another gene that we found. Again, association with breast cancer and lump and mass breast abnormal findings and non-Hodgkin's lymphoma. So all these seems to make a lot of sense for our breast cancer collaborators. So we still need to validate this as just an example of other hits that we've had. We've had multiple of those. And then there's the question of course, structure. Is the structure forming? Coming back full circle, sort of where we started. So this is work, collaborative work with Dan Dominguez from UNC and his student Brian. So they use, again, circular dichorism, and you can see 2 wild types that were identified in EPN3, and you can see the profiles in different temperatures that indicates G4 formation. And interestingly, the one on the right that you're seeing here is a shortened version of one of them, where we're basically shortening it, and then the expectation is that it will form stronger connections, and that's what we see, because the first row on the top is for using potassium. That is a stronger connection, and the bottom row is for lithium, so it's weaker. Only when we shorten it, we start seeing the signal there for the formation. So what else? We don't want to do more additional experiments for validations, both for the effects and for the structure. We can also not actually distinguish here between effects on translation, right, or stability, because we're just measuring protein, stable protein levels. So that's another interesting question to follow. We're also following up on model interpretability. We've implemented a version for those transformers. Nate from the group did this for enhanced integrated gradients. That works really well, and we're still analyzing and want to validate the results for that, so that's ongoing. And then we also build a web tool for this, so anybody can plug in any sequence and put the power of the model for use. So I'll just thank all the people who did the terrific work here. Farica, first and foremost, Danielle, who did the experiment, Nate and San, who helped with the web tool and the interpretation. Kate Nathanzone is our collaborator in breast cancer. Dan and his student Brian from UNC helped with the circular dichorism. This is all the lab, the funding, and thank you for your time.
Thank you. Please.
Okay, thanks. So regarding the subtype classification, I wonder how you define the problem and where you got the labels from, because if it's based on a regular expression, then you don't need the machine learning form, or is it from an experiment that you get?
So RG4Seeker, the paper where they sort of improved their labeling, they also label the types that they find, and we basically fed the algorithms those labels.
Okay, I wonder if you can just take the procedure from RG4Seeker and you get a classification. Would that work?
So maybe. We haven't tried that, but I think there is, what we're trying to see is what are we able to learn at the same time, and whether that will give us some hints about interpretability, which is that other section.
Really nice work. Can you comment on the effects on expression? So the luciferase assay tells you about level of protein, right? And are they always negative in their action? I mean, do they
No, so I
repress, or
showed
you have
variants
both, or-
breaking or forming, right? And,
Right.
And so you could potentially have effects in either directions. But for expression, luciferase don't measure it. What I didn't show is a point well made, especially for 3' UTRs, right, where you would expect. So what we showed in the original paper that we published, and there, if the variant is common enough, you can do a little specific expression, and we've done that for a whole bunch, and you see very clear signal in some cases. But yeah.
So they just show up as EQTLs or?
Yes,
Yeah,
and
that's what
in that paper
In
that
that-
I cited, you can see some
and-
nice examples that are also GWAS hits.
I see. And when, but in general, like, is the presence of the RG4 always changing expression in the same
Oh.
direction, I guess?
Good question. I don't remember what the answer is. I'm sorry.
With the examples that you do know, is it usually negative, or is there?
I think it's negative. I
Negative.
think it was negative,
Okay.
but yeah.
Okay, thanks.
Sorry.
Thank you. For long G4s, did you check if it's only the G tracks that are important for the prediction, or could the linkers be important, and maybe you could pick up some RNA binding protein binding sites?
Yes. So that relates to Yaron's question, right? So once you have those subtypes and you have the longer sequences, can you find, and that relates to Nate's work. So we do have some signals there, but since I don't have like solid results that I, can present with yeah. So yeah, but it's a very good point. Yeah.